Welcome to the Deep Dive. I am so glad you could join us today.
Yeah, thanks for having me. It's great to be here.
So today we have a really unique mission for you. We are diving into basic poultry science.
Right, and specifically, we are looking at a highly infectious disease. It is called Infectious Bursal Disease, or IBD for short.
Exactly. And we are also going to examine a very specific preventative tool. It's a product from Elanco called AviPro IBD Extreme.
Yeah, the sources we have today go into incredible detail about how this virus works and, how this specific vaccine is engineered to fight it.
But before we jump in, I want to talk directly to you, the listener, because today's format is a little bit different.
Right, we are doing something special today.
Yeah. This deep dive is specifically designed for a beginner, someone who is learning professional poultry vocabulary.
So because of that, we are going to be speaking at a moderately slow pace.
And we are going to use short, clear sentences.
I mean, poultry farming is a highly scientific field. It really is.
It's basically applied biology on a massive scale.
Exactly. And understanding the language, the specific vocabulary, is the absolute first step to mastering the practice.
So throughout this deep dive, we are going to introduce 12 essential technical terms.
12 key words that you will hear every day on a modern poultry farm.
And we will define each of those terms in simple English.
We will repeat them naturally in the conversation.
And we will give you a practical farming example for every single one.
And here is the most important part for you, the listener. We are going to leave brief pauses after those important example sentences.
Right. We want you to practice something called shadowing.
Shadowing is when you repeat the sentence out loud right after you hear it.
It is a fantastic way to build your vocabulary. So when you hear us slow down and give an example, just say it right out loud wherever you are.
It really works. So where should we start?
I think we have to start by understanding the enemy. Before we can talk about a vaccine, we have to look at the battlefield.
Right. We need to go back in time a bit.
Yeah. Back to 1962, there was a mysterious illness sweeping through poultry farms.
This was in a small town called Gumboro. It's in Delaware.
Young chickens were just dying at these alarming rates.
Yeah.
And farmers were completely panicked.
I mean, you can imagine the veterinarians came in and they opened up these birds to see what was going wrong.
And when they looked inside, they found that the kidneys were just massively enlarged.
Yeah, severely damaged.
Yeah.
So naturally, based on what they saw, they called it avian nephrosis.
Nephrosis meaning kidney disease.
Exactly. But they were entirely wrong.
They were looking at the wrong thing.
Yeah. The kidneys were just collateral damage. The real target was something far more insidious.
And the pathogen behind it was basically wearing a biological suit of armor.
The story of what happened in Gumboro and what continues to happen on farms across the entire planet today is a masterclass in viral evolution.
It really is. If you follow agriculture at all, or global food supply chains, you know how hard this is.
Managing disease in a flock of 50,000 birds is just a monumental task.
So let's get into our very first vocabulary word for today. This is the word we are talking about. The word is broiler.
Broiler.
A broiler is a chicken that is raised specifically for meat.
Right, not for laying eggs, but for meat.
Here is your practical farming example. Get ready to shadow this sentence.
The farmer checks the water lines to make sure every broiler is drinking enough today.
Go ahead and repeat that. The farmer checks the water lines to make sure every broiler is drinking enough today.
Excellent. So that is the host animal, the broiler. Now let's talk about the invader.
Which brings us to our second word, pathogen.
Pathogen.
A pathogen is a germ or a virus that causes disease.
It is any microscopic organism that makes the animal sick.
Here is your farming example to practice.
Right.
We use strong disinfectants to clear any pathogen from the barn.
Go ahead and try it. We use strong disinfectants to clear any pathogen from the barn.
Great job. So we have the broiler and we have the pathogen.
And to really understand why a product like AviPro is necessary, we have to rethink how we view the chicken's immune system.
Because it is very different from ours, right?
Completely different. When human beings get an infection, our immune response relies heavily on our lymph nodes.
And our bone marrow.
Exactly. But birds are fundamentally different. They do not have lymph nodes in the way mammals do.
They rely on a highly specialized central organ instead.
Right. An organ specifically designed to produce their B lymphocytes.
Those are their antibody-producing cells, for anyone taking notes.
Yeah. And this specific organ is crucial to our third vocabulary word.
Let's unpack this because the biology here is fascinating. The word is bursa of Fabricius.
Bursa of Fabricius.
It is a special organ in chickens that makes immune cells.
It is the absolute command center for a young bird's immune system.
Here's your farming example to shadow. A healthy bursa of Fabricius means the chicken can fight off infections.
Let's hear you say it. A healthy bursa Fabricius means the chicken can fight off infections.
Perfect.
Yeah.
So tell us a little bit more about this organ. Where is it?
Well, it is a small sac-like organ. It is located right near the bird's cloaca.
And it has a pretty interesting history, right?
Yeah, it was actually discovered way back in the 16th century by an anatomist named Girolamo Fabricius.
Which is obviously where it gets the name.
Right. But for centuries, nobody actually knew what it did.
It was just a biological mystery.
Exactly. It wasn't until the 1950s that scientists finally realized it is the training camp for the immune system.
If a broiler has a healthy bursa, it can generate the antibodies needed to survive.
Because a barn environment is full of pathogens every single day.
And here is the truly sinister genius of the IBD virus.
Right. It doesn't just attack the bird's respiratory system.
Or its digestive tract.
No. It acts kind of like HIV does in humans. It directly targets the immune system itself.
The virus specifically seeks out those actively dividing B cells inside the bursa of Fabricius.
It gets inside them, it hijacks their cellular machinery to replicate itself.
And it destroys the cells entirely in the process.
It just wipes them out.
So going back to those scientists in the 1960s who saw kidney damage.
Yeah, the virus was not targeting the kidneys at all.
Not even a little bit.
The kidney damage was just a secondary symptom. The birds were suffering from severe dehydration.
And massive blockages from the overwhelming viral load, right?
Exactly. The virus was always aiming for the bursa.
I want to zoom in on the physical virus itself for a minute.
Okay, let's do it.
Because the sources we are looking at detail its physical structure.
Huh.
And it really explains why this pathogen is such a nightmare for farmers to eradicate.
It is a very stubborn virus.
It belongs to a family called Burniviridae.
Right, Burniviridae. And that name actually tells you a lot about its genetics.
How so?
Well, bi means two, and RNA stands for its genetic material.
So it has two distinct segments of double-stranded RNA.
Yes. But beyond its genetic code, its physical structure is what makes it a permanent resident on poultry farms.
The sources mention that it is a non-enveloped virus.
Right. Non-enveloped. This is a crucial detail.
Let's unpack that. Being non-enveloped is like wearing a tough suit of armor, isn't it?
That is the perfect analogy.
Because with enveloped viruses, they have this soft, fatty outer layer, a lipid membrane.
And because that envelope is basically made of fat, you can destroy it pretty easily.
Just with basic soap and warm water, or standard alcohol disinfectants.
Right. A lipid envelope is like wearing a soft fleece jacket. It tears easily.
But the IBD virus does not have that soft jacket.
I know. By contrast, being a non-enveloped virus is like wearing a solid, interlocking suit of titanium armor.
It is encased in a highly stable, rigid protein shell.
Called a capsid, and that capsid protects it from almost everything.
So let's put this in a real-world scenario. A farmer has a sick flock. They remove all the birds from the barn.
Okay.
They scrub the floors. They wash the walls with industrial disinfectant.
Yeah, they do a deep clean.
And then they bring in a brand new, healthy flock of broilers. Is the virus just sitting there totally unfazed?
In a lot of cases, yes. It is incredibly persistent.
That is terrifying for a farmer.
It can survive in the environment for a very long time. In the dust, in the litter on the floor, in the water lines.
The sources even mention insect vectors, like darkling beetles.
Yeah, darkling beetles live in the barn. They eat the dust. The virus survives right inside their gut.
And then the new chickens come in and eat the beetles.
Exactly. And the cycle starts all over again. The virus can survive like this for months, sometimes over a year.
Because that protein armor protects it from heat, from extreme pH changes, and from standard quaternary ammonium disinfectants.
This is exactly why IBD is considered endemic in almost every major poultry-producing region in the world.
It is just a constant, unavoidable environmental pressure.
Which brings us to how the virus actually manifests when it finally hits a flock.
Right. Our sources outline that there are different serotypes.
Yeah, there is serotype 2, which is actually harmless. It does not cause clinical disease.
But serotype 1 is the dangerous one.
Very dangerous. And within serotype 1, we have two very different battlefronts.
Classical strains and variant strains.
Let's look at the classical strains first. These are the ones that cause the acute fiery outbreaks.
When a flock of susceptible birds encounters a classical strain, the onset is just explosive.
And this usually happens when the birds are between 3 and 6 weeks of age.
Which introduces our next important vocabulary word, word number 4, Morbidity.
Morbidity.
Morbidity is the rate or percentage of illness in a flock.
Right. It is not the percentage that die, but the percentage that actually get sick.
Here is your farming example to shadow. Because the virus spreads fast, the morbidity in the flock reached nearly 100%.
Your turn to practice. Because the virus spreads fast, the morbidity in the flock reached nearly 100%.
Great. Now 100% morbidity. That is just staggering to think about.
It really is. A barn could hold 50,000 broilers.
And within 48 hours, virtually every single one of them is infected.
Because the incubation period is incredibly short. It is just 2 to 3 days.
So if you walk into a house hit by classical IBD, what does it actually look like?
The clinical signs are obvious and honestly heartbreaking.
The sources say the birds exhibit severe anorexia.
Yeah, they completely stop eating and drinking.
And they have this hunched, depressed posture.
With severely ruffled feathers, they puff up to try to conserve body heat.
Because they are running a massive fever.
Exactly. You will also see whitish, watery diarrhea and severe muscle tremors.
And what about the mortality rate? How many birds actually die?
It spikes dramatically.
Yeah.
Especially with highly virulent strains.
The ones virologists call IBDV.
Right, very virulent IBDV. With those strains, you can see up to 20% of the flock die in a matter of days.
The mortality curve goes straight up, peaks around day 4, and then drops back down almost as quickly as it started.
It is a devastating direct economic loss. If you lose a fifth of your flock, your profit margins for that cycle are just gone.
But the literature suggests something really counterintuitive here.
What's that?
It suggests that the variant strains, the ones that do not cause that massive wave of death, are actually worse for the global industry.
Yes, that is absolutely true.
But how is a virus that doesn't kill the bird worse than one that does?
Because it is a silent thief. Variant strains do not cause the acute visible inflammation that classical strains do.
There is no spiking mortality curve.
No, a farmer might walk through the barn and think the flock looks generally okay.
But inside the bird, something terrible is happening.
The variant strain is causing severe bursal atrophy. The bursa of Fabricius shrinks, it wastes away, and it stops producing B cells entirely.
Which triggers our next major concept, word number 5, immunosuppression.
Immunosuppression.
This means the immune system is too weak to fight off diseases.
It is effectively deleted.
Here is your farming example to shadow. The main danger of the virus is the immunosuppression it causes in young birds.
Go ahead and say it. The main danger of the virus is the immunosuppression it causes in young birds.
Excellent. So immunosuppression is the ultimate danger here.
Yes. Without a functioning bursa, the bird cannot mount an immune response to any other pathogen in the environment.
And a poultry barn is full of opportunistic bacteria.
Things like E. coli are everywhere.
So what does this silent immunosuppression mean for the farmer's bottom line?
It means indirect losses that bleed the farm dry over weeks.
Because the birds are suffering from chronic secondary infections, right?
Exactly. Respiratory and digestive infections. Because they are sick, they don't convert their feed into muscle efficiently.
Which destroys the farm's feed conversion ratio.
And because they are constantly fighting off bacterial infections, the farmer is forced to use higher levels of antibiotics.
Which goes completely against modern agricultural goals of reducing antibiotic usage.
It is a vicious cycle. And the final insult happens at the processing plant.
When the birds are sent to the slaughterhouse.
Right. When those surviving, chronically ill birds reach the plant, the inspectors examine them.
And they find lesions or septicemia or air sacculitis.
And those carcasses are condemned. They are thrown out.
They are not allowed into the human food supply.
So the farmer paid to feed and house that bird for 6 weeks, only to get zero return on investment.
The economic ripple effect of this silent immunosuppression is literally billions of dollars globally.
It is an invisible disaster.
So how do you even confirm that IBD is the ghost haunting your farm? You can't just guess.
No, you need a definitive diagnosis. It starts with the clinical presentation, but it moves very quickly to the necropsy table.
Which brings us to word number 6, necropsy.
Necropsy.
A necropsy is an animal autopsy to find out why it died or got sick.
It allows the veterinarian to look for physical clues inside the body.
Here is the example sentence for you to shadow. The veterinarian performed a necropsy to look for hemorrhages in the thigh muscles.
Give it a try. The veterinarian performed a necropsy to look for hemorrhages in the thigh muscles.
Good job. So they are opening the bird up and looking straight at the bursa.
Right. With a classical strain, the bursa might be swollen to twice its normal size.
The sources say it can be covered in a yellowish jelly-like fluid.
Or even hemorrhagic, turning dark red or black from internal bleeding.
And they also look for those pinpoint hemorrhages in the muscles and the proventriculus, right?
Yes, the glandular stomach of the bird.
But if it is a variant strain, they're looking for the exact opposite.
Exactly, a bursa that has shrunk down to the size of a pea.
But gross necropsy only tells you so much. You just see the damage.
To truly know what armor the enemy is wearing, you have to take tissue samples.
And send them to a lab.
Right. They use serology, like ELISA testing, to look at antibody levels in the blood.
But the sources point out that the gold standard today is molecular testing.
Yes. PCR, polymerase chain reaction.
PCR is what we use to look at the literal genetic code of the virus.
PCR and genetic sequencing tell the farmer exactly which strain of IBD is present on the farm.
Is it classical? Is it a variant? Is it a very virulent strain?
Once you have that genetic blueprint, you can formulate a strategy.
And that brings us to the counterattack. The solution.
Because you have an armored, highly persistent virus that either kills the bird outright or deletes its immune system.
And you can't clean it away with soap. The only way to win is to prepare the bird's immune system before the virus strikes.
This is where we dive into the specific tool outlined in our sources, Elanco's AviPro IBD Extreme.
It is a really fascinating piece of biotechnology.
But delivering a fragile, living biological product to millions of chickens safely is a massive logistical hurdle.
Let's talk about that logistics piece.
Yeah.
Because you can't just ship liquid virus in a cardboard box.
No, it would degrade immediately.
So the AviPro vaccine arrives at the farm in a very specific format, which is word number 7. Lyophilisate.
Lyophilisate.
A lyophilisate is a freeze-dried powder. It is a common way to store vaccines safely.
It keeps the virus stable during shipping.
Here's your example to shadow. The vaccine arrives at the farm as a lyophilisate in a small glass vial.
Let's hear you say it. The vaccine arrives at the farm as a lyophilisate in a small glass vial.
Perfect. So explain how they actually make this freeze-dried powder.
It is a brilliant preservation method called sublimation.
Sublimation, right?
They freeze the biological material, and then they reduce the surrounding pressure.
Which allows the frozen water to transition directly from a solid ice phase to a gas phase.
Exactly. Skipping the liquid phase entirely.
It essentially puts the live vaccine virus into suspended animation.
It stabilizes it so it can survive storage in a standard refrigerator without dying.
But obviously, a broiler chicken cannot consume a dry powder.
No. Which brings up the next step for the farmer.
Word number 8, reconstitution.
Reconstitution.
This means mixing a dry powder with a liquid so it can be used.
It wakes the virus back up.
Here is the shadowing example. After reconstitution with water, the vaccine is ready to be given to the birds.
Try it out loud. After reconstitution with water, the vaccine is ready to be given to the birds.
Great. Now, I imagine reconstitution is not as simple as just dumping the powder into a plastic bucket.
Far from it. Live vaccines are incredibly sensitive.
Because it's a living thing.
Right. If the farm's water has high levels of chlorine from cleaning, it will kill the vaccine.
Or if the pH is too acidic.
Or even if the water pipes are just hot from the summer sun, it can kill the vaccine virus instantly.
So how do farmers protect it?
They often have to add stabilizers to the water first, like skim milk powder or specific blue dyes.
Blue dye. Why blue dye?
Well, the dye helps neutralize the chlorine, but it also serves a visual purpose.
It temporarily turns the bird's tongues blue, doesn't it?
Exactly. So the farmer can walk through the flock later and visually confirm, yes, that bird actually drank the vaccine.
That is so practical. Okay, so we have reconstituted the vaccine. But how do we know it is actually strong enough?
That is a great question.
With a chemical drug, you measure it in milligrams. You know exactly how much chemical is in the pill.
But you can't weigh a living, replicating virus like that.
Right. How do you measure biological chaos?
You measure its infectivity. And in virology, you will see a very specific measurement on the label.
Which is our 9th vocabulary word. It is a bit complex, but we will break it down. The term is EID50. That stands for 50% embryo infective dose. It is the dose of a virus needed to infect half of the test eggs.
It guarantees the potency of the vaccine.
Here's the example sentence for you to shadow. We ensure high quality because one dose contains at least 10 to the power of 1.5 EID50.
Let's hear you say that complex sentence. We ensure high quality because one dose contains at least 10 to the power of 1.5 EID50.
Awesome job. So explain to us exactly how they calculate this EID50.
It is a statistical measurement. To find it, scientists take embryonated chicken eggs.
Eggs that have a live, developing chick embryo inside.
Right. They inject a highly diluted sample of the vaccine into a batch of these eggs.
And then they do a slightly less diluted sample into another batch.
And so on. The EID50 is the exact dilution dose required to successfully infect exactly 50% of the embryos in that test batch.
So it is a standardized mathematical baseline.
Yes. It ensures that when a farmer reconstitutes a 5,000-dose vial into their water lines, there are enough viable viral particles to establish immunity in 5,000 individual birds.
Okay, let's look at what is actually inside that vial of AB Pro IBD Extreme.
The sources categorize it as a live intermediate plus vaccine,
And it utilizes a specific strain called V217.
which is derived from a very famous historical strain called Winterfield 2512.
But I want to focus on that phrase, intermediate plus. What does that actually mean in the context of a live vaccine?
To understand that, you have to understand the tightrope walk of poultry vaccinology.
It is a balancing act.
Totally. If you use a mild vaccine strain, it is very safe. It won't cause any damage to the bursa.
But because it is so weak, it might not generate a strong enough immune response.
Exactly. It gets easily wiped out by the bird's natural defenses before it can even work.
On the flip side, if you use a hot or highly virulent vaccine strain, it will definitely trigger a massive immune response.
But it might actually cause the exact bursal atrophy you are trying to prevent.
You've essentially just given the bird the disease.
Precisely. So scientists created intermediate strains that try to balance safety and efficacy.
But even regular intermediate strains had a weakness, which we will get to in just a second.
So they developed intermediate plus strains, like Avi Pro.
These are slightly more invasive than standard intermediate strains.
They replicate a bit stronger in the bursa, which means they stimulate a much more robust, profound immune response.
But they are still attenuated or weakened enough that they do not cause permanent bursal damage or clinical disease.
And they need that extra strength, that plus because of the biggest roadblock in all of pediatric immunology.
This applies whether you're talking about humans or chickens, and that roadblock is the mother.
Yes. Which leads us to vocabulary word number 10, maternal antibodies.
Maternal antibodies, often abbreviated as MDA.
It is the natural immunity a baby chick gets from its mother hen.
Here is your shadowing example. We have to time the vaccination perfectly so it works when the maternal antibodies drop.
Let's practice that. We have to time the vaccination perfectly so it works when the maternal antibodies drop.
Great. So let's walk through how this maternal antibody transfer works, because it is a beautiful piece of natural engineering.
It really is. When a mother hen, a broiler breeder, is exposed to IBD in her life, or if she receives an injected vaccine, she develops massive amounts of circulating antibodies in her blood.
And nature knows that her chicks are going to be born into a dangerous barn environment.
So during the formation of the egg, the hen physically transfers a massive payload of her own IBD antibodies directly into the egg yolk.
As the chick embryo develops, it absorbs that yolk.
And those maternal antibodies enter the chick's bloodstream.
So when that chick hatches, it is already wearing its mother's biological armor.
It has what we call passive immunity.
It's an amazing system for the first week or two of life. If the field virus attacks a 3-day-old chick, the mother's antibodies neutralize it immediately.
But here's the fatal flaw. Passive immunity does not last forever.
The maternal antibodies degrade over time.
Their half-life in a chick is roughly 3 to 5 days,
So by day 14 or day 18, the mother's armor is basically gone,
leaving the chick completely naked and vulnerable to the virus in the barn.
which means the farmer has to step in and vaccinate the chick so its own immune system can take over.
It needs to produce its own active antibodies.
But here is the paradox. Here is where it gets incredibly tricky for the farmer.
A live vaccine like AviPro is, by definition, a virus.
It's a safe, weakened virus, but to the chick's immune system, it looks identical to the real threat.
So if a farmer vaccinates a chick while it still has high levels of maternal antibodies,
The mother's antibodies will do exactly what they were designed to do.
they will attack the vaccine virus, neutralize it, and clear it from the bird's system before the vaccine ever reaches the bursa of Fabricius.
The vaccine is completely wasted.
It is a phenomenon called vaccine interference.
So if you vaccinate too early, the mother's immunity kills the vaccine, and the chick remains unprotected later.
But if you wait too long for the maternal antibodies to drop to zero, there is a massive window of susceptibility where the wild field virus can infect the flock and cause devastation.
How do you thread that needle? How do you protect the bird without the mother fighting you?
You need a vaccine with what we call a high breakthrough titer.
This is the exact reason AviPro IBD Extreme uses that intermediate plus V217 strain.
Right. Standard mild or intermediate vaccines get easily neutralized by even moderate levels of maternal antibodies.
You have to wait until the birds are almost 3 weeks old to use them safely, which is very risky.
But AviPro's strain is robust enough that it can actively break through higher levels of maternal immunity.
It can punch through the mother's declining defenses earlier in the chick's life.
This gives the farmer incredible flexibility. They don't have to wait until the birds are 21 days old and highly vulnerable.
They can administer the vaccine earlier?
Yeah.
The sources mention application through drinking water, spray, or eye drop.
Right. And it comes in various pack sizes, 500, 1,000, all the way up to 10,000 doses.
So it fits different farm sizes perfectly.
And by giving it earlier, you establish high levels of active protective antibodies much sooner.
Protecting the zootechnical performance of the flock without leaving a dangerous gap in protection.
And the way this vaccine actually confers that protection inside the bird is through a brilliant biological mechanism.
Which is our 11th vocabulary word, competitive exclusion.
Competitive exclusion.
This is when a safe, helpful vaccine virus occupies the cells first, blocking the bad field virus from attaching.
It is literally a competition for space.
Here's the example sentence to shadow. The vaccine protects the bursa through competitive exclusion.
Let's practice that. The vaccine protects the bursa through competitive exclusion.
Awesome. So let's explain this concept because it is one of the most elegant concepts in immunology.
Let's think back to the anatomy of the bursa.
Right.
The IBD virus, whether it is the dangerous field strain or the safe vaccine strain, has to physically bind to specific cellular receptors inside the bursa of Fabricius to replicate.
Those receptors are basically like parking spaces.
Exactly. There are only so many of them available on the surface of the cells.
Okay, so the farmer administers the AviPro vaccine via the drinking water.
The live, safe V217 vaccine strain travels through the bird's body to the bursa.
It finds those receptor parking spaces, pulls in, and sets up shop.
It begins replicating gently. It stimulates the immune system, but it causes no permanent harm.
And because the vaccine virus has already occupied those specific cellular receptors, what happens when the lethal wild field virus enters the barn 2 weeks later?
The dangerous field virus enters the bird, travels to the bursa, and finds the parking lot completely full.
There are no available receptors.
The field virus simply cannot attach to the cells.
If it cannot attach, it cannot enter the cell.
If it cannot enter, it cannot replicate.
And if it cannot replicate, it just gets flushed out of the bird system entirely.
The vaccine competitively excludes the pathogen from colonizing the target organ.
It is essentially a biological blockade.
But, as with everything in science, theoretical mechanisms and parking lot analogies are great, but they don't mean anything until they are tested against the chaos of the real world.
Because viruses don't just sit still and let vaccines win.
No, they certainly do not.
RNA viruses like IBDV are notoriously prone to mutation.
Because their RNA polymerase, the enzyme that copies their genetic code, lacks a proofreading mechanism.
So they make mistakes every time they replicate.
Most of those mistakes are fatal to the virus. But occasionally, a mistake creates a new trait that helps the virus survive.
And because the IBD virus has a segmented genome, remember, it has 2 distinct pieces of RNA, segment A and segment B, it can do something even more radical than a random mutation.
It can undergo reassortment. Which brings us to our final vocabulary word, word number 12.
Reassortant strain.
Reassortant strain.
A reassortant strain is a virus that has swapped genetic parts with another virus to create a new, tricky version of itself.
It is viral shuffling.
Here is your final shadowing example. The laboratory confirmed that the farm was attacked by a new reassortant strain.
Let's hear you say it. The laboratory confirmed that the farm was attacked by a new reassortant strain.
Excellent job. You have practiced all 12 words. Now let's talk about how this reassortment actually happens in nature.
If a single chicken cell is unlucky enough to be infected by 2 completely different strains of the IBD virus at the exact same time.
Say, a highly lethal strain and a mild immunosuppressive strain.
Right. Those viruses are replicating their RNA segments in the same cellular soup.
When the new viruses are being assembled, they can accidentally package segment A from the lethal strain and segment B from the mild strain.
They swap genetic cards, and out pops a reassortant strain.
A completely novel virus that the world has never seen before, with a Frankenstein combination of traits.
This is exactly what causes massive global pandemics in human influenza, by the way.
And it is exactly what happened in the European poultry industry recently, which is the final and most compelling piece of data in our sources.
The European Challenge Study.
Let's set up the problem first. For years, a highly virulent classical strain known as DV86 was the primary terror in Europe.
It caused high mortality. Everyone knew what it looked like.
But starting around 2016, veterinarians in Western, Central, and Northern Europe noticed something weird.
DV86 was vanishing. They weren't finding it on PCR tests anymore.
But the flocks weren't performing well. The feed conversions were terrible. Secondary infections were up.
The farmers knew something was deeply wrong, but there were no dead birds piling up to signal an IBD outbreak.
Because a new reassortant strain had taken over.
This new virus had swapped its genetics so that it hardly caused any outward clinical signs. The mortality was effectively zero.
But inside the bird, this reassortant strain was ruthlessly efficient at causing severe permanent bursal atrophy.
It was the ultimate stealth bomber. It flew entirely under the radar of the farmer, quietly destroying the immune systems of millions of birds.
It is a terrifying evolutionary strategy. If a virus kills its host quickly, it limits its own ability to spread.
But if a virus keeps the host alive, looking totally normal, while it multiplies by the billions and sheds into the environment.
That is a wildly successful pathogen.
So the critical question for Elanco was this. Could AVPRO-IBD Extreme, a vaccine based on a historical strain, protect against this brand new, highly stealthy European reassortant strain?
To find out, they set up a highly controlled challenge study. Let's walk through the methodology.
The way these trials are designed is really fascinating.
They started with 25 SPF broilers.
SPF stands for specific pathogen-free.
These are essentially bubble boy chickens, right?
Exactly. They are raised in heavily filtered, highly biosecure isolation units.
They have never been exposed to any viruses, any bacteria, or any vaccines in their entire lives.
Their immune systems are a completely blank slate.
They divided these 25 pristine birds randomly into 3 distinct groups.
Group 1 was the test group. This was 10 birds.
At day 14 of their lives, these birds were given the AVPRO vaccine orally.
Then we have Group 2. This is the vulnerable group, also 10 birds.
They received no vaccine at all. They were left completely unprotected.
And Group 3 was the negative control group, just 5 birds.
They received no vaccine, and they would receive no virus later.
They were just allowed to live in perfect isolation, so the scientists had a baseline of what a perfectly healthy, untouched bursa looks like.
The timeline of the study is crucial here. At day 14, Group 1 gets the vaccine.
1 week later, at day 21, the challenge occurs.
Challenge being the polite scientific term for intentionally infecting the birds with a dangerous disease.
Yes. The scientists took that sneaky, highly damaging European reassortant field strain.
And they administered it directly to the birds in Group 1 and Group 2.
Via oculonasal drops, literally dropping the live wild virus right into their eyes and nostrils to guarantee a massive respiratory and mucosal exposure.
So Group 1 has had the vaccine for exactly 1 week. Is 1 week enough time for the V217 strain to establish competitive exclusion in the bursa?
And Group 2 has no protection at all.
5 days after the challenge, at day 26 of the birds' lives, the scientists concluded the trial.
They humanely sacrificed the birds and conducted necropsies, focusing entirely on the bursa of Fabricius.
The first metric they looked at was the BLS.
Bursal lesion score.
This is a quantified visual assessment of the physical damage to the organ.
The scale generally goes from 0, meaning perfectly healthy, up to higher numbers indicating severe necrosis, atrophy, and hemorrhage.
Let's look at the results. In Group 3, our negative controls who lived in the bubble, the score was 0, perfect health.
Which is exactly what you would expect.
In Group 2, the unvaccinated birds that were challenged with the reassortant strain, the bursal lesion score was a highly damaging 2.8.
The virus had absolutely ravaged their immune organs in just 5 days. The atrophy and destruction were severe.
If these birds had been in a commercial barn, they would be globally immunosuppressed for the rest of their lives.
But then we look at Group 1, the birds that received AVPRO IBD Extreme just 7 days before being blasted with the wild virus.
Their bursal lesion score was 0.4.
Barely above absolute 0, the physical protection was staggering.
The vaccine prevented almost all the severe, long-lasting morphological damage to the organ.
But the scientists did not stop at just looking at the organ visually. Visual scores are subjective.
Right, they went down to the molecular level.
They extracted tissue from those bursas and ran PCR tests and full genetic sequencing.
They wanted to know exactly whose RNA was currently residing inside those cells.
This is where the concept of competitive exclusion gets proven beyond a shadow of a doubt.
Let's hear the data.
In Group 2, the unvaccinated birds, the PCR results showed 98.1% homology with the dangerous challenge strain.
The enemy had successfully invaded, colonized, and taken over the entire organ.
And in Group 1, the vaccinated birds.
The sequencing showed 100% homology with the Avitro vaccine strain.
The safe V217 strain was sitting comfortably in all the cells.
And what percentage of the reassortant challenge strain did they find in Group 1?
0%. Absolute zero.
That is incredible.
The vaccine had prevented the multiplication of the new field strain by 100%.
The wild virus entered the bird's body, traveled to the bursa, and found every single parking space occupied by the vaccine.
It couldn't attach, it couldn't replicate. It was completely excluded and flushed from the bird system without leaving a single trace.
Within just 1 week post-vaccination, the door was entirely shut.
The bird's immune system remained completely intact, ready to mount responses to any other environmental pathogens.
Ensuring that the broiler could grow efficiently and safely reach its target weight.
It is a perfect synthesis of applied immunology. We take a highly robust, intermediate plus strain that can punch through maternal antibodies early.
You allow it to colonize the bursa safely.
And by doing so, you build an impenetrable wall against even the most highly mutated, stealthy field strains nature can throw at you.
We have covered an immense amount of ground today.
We really have.
From the historical mysteries of avian nephrosis in the 1960s to the molecular armor of the Bernaviridae family,
We explored the devastating economic reality of silent immunosuppression.
the complex mathematics of viral dosing with EID50,
And the beautiful, brutal competition for cellular receptors inside a chicken's immune system.
The overarching takeaway from our sources today is that poultry farming is not just putting feed and water in a barn.
No, it is high-level, cutting-edge biological warfare.
Pathogens, like the reassortant IBD strain, are constantly adapting, finding new ways to circumvent host defenses without killing the host too quickly.
It is a constant battle.
Now, before we let you go, as promised, let's do a rapid but clear recap of our 12 key vocabulary words.
See if you remember what they mean.
1. Broiler.
2. Pathogen.
3. A bursa Fabricius.
4. Morbidity.
5.
Autopsy.
7. Lyophilizate.
8. Reconstitution.
9.
The IB50.
10. Maternal antibodies.
11. Competitive exclusion.
And 12. Reassortant strain.
You know, before we wrap up this deep dive entirely, I want to leave you, the listener, with a final thought to mull over.
A provocative thought.
We saw today how this European reassortant strain brilliantly altered its genetic code to drop its mortality rate to 0, becoming a silent, invisible destroyer of immune systems.
It completely bypassed the farmer's visual detection.
It is a chilling reminder of viral intelligence.
Exactly. So here's the question to explore on your own. If RNA viruses like IBD are constantly mutating, constantly shuffling their genetic decks to evade detection and exploit new vulnerabilities,
How close are we to a reality where agricultural vaccines must be updated and reformulated every single year?
exactly the same way we have to update the human flu shot. We rely on these massive flocks for global food security, and the arms race between viral evolution and human biotechnology is happening right now in barns all over the world every single day.
The virus never stops adapting. The science can't either.
It really makes you look at a simple chicken breast in the grocery store in a completely new light. Thank you for joining us on this journey into the microscopic battlegrounds of agriculture. Keep questioning, keep digging, and we will see you on the next deep dive.